Welcome to lecture on sequence comparison and clustering. Myself Dr. D.S. V.Z.K. Kaladar, Associate Professor, Microbiology and Bioinformatics, UTD, Atal Bihari Vajpayee University, Bilaspur, Chhattisgarh. Sequence comparison. In bioinformatics, a sequence alignment is a way of arranging the sequences of DNA, RNA or protein to identify regions of similarity that may be consequence of functional, structural and evolutionary relationships between the sequences. When we compare sequences, we take two or pairwise or more, that is multiple strings of letters, either nucleotides or amino acids, and align them. Differences in the sequence can be caused by deletions or insertions in the DNA or by point mutations. Sequence comparison techniques are two types. One is the pairwise alignment, dot plot, local alignment or Smith-Waterman algorithm, global alignment or Needleman and Wunsch algorithm. Second one is multiple alignment, hierarchic method. Rather than struggling to find the optimal alignment, lot of time can be saved by employing hierarchic algorithms. Execution time is better and faster, may completely miss the optimal alignment, that is in FASTA and BLAST. In pairwise sequence comparison or pairwise alignment, first one is dot plot. Put one sequence on top, the other on the side. Where residues are identical, put a dot. Diagonal lines of dots show similarities. This is one of the examples for comparison: sperm whale myoglobin and human myoglobin. Coming to the examples for making dot plot. If you take the two examples, one is the palindromic sequence, another is the same sequence. In palindromic sequence, the alignment will be AATT, and with the gaps in the second one, the after two gaps, TTAA, and in the case of the same sequences, TATAAT, TATAAT. Coming to Needleman and Wunsch algorithm. The Needleman and Wunsch algorithm is an algorithm used in bioinformatics to align protein or nucleotide sequences. The global sequence alignment method or Needleman and Wunsch algorithm is the first to apply dynamic programming techniques to biological sequence analysis, which explores all possible alignments and chooses the best one. This is the website for the Needleman and Wunsch algorithm. The formula for Needleman and Wunsch algorithm is Ti comma j is equal to max of Ti minus 1 comma j minus 1 plus dou S1 I S2 j. And in the second line, Ti minus 1 comma j plus gap penalty. In the third, Ti comma j minus 1 plus gap penalty. Coming to the steps involved in global alignment or Needleman and Wunsch algorithm. The first step is initialization. Create matrix with m plus 1 columns and n plus 1 rows, m sequence 1 and n as sequence 2. The second step is matrix fill. Fill first column and last row with gap value. If there is a match, give plus 1, mismatch minus 1 and gap as minus 2. Insert values by adding gap values with decide box. So hence in the 0, we will write as 0, minus 2, minus 4, minus 6, minus 8. And in the vertically also 0, minus 2, minus 4, minus 6, minus 8. Coming to the third step, scoring. So max value of box beside plus gap value of box bottom minus gap value, diagonal value plus mismatch or mismatch. If there is a match, give plus 1, mismatch as minus 1 and gap as minus 2. Coming to this example, G and G. If you take this one, so the first one is value beside the box is minus 2 plus the gap value, that is minus 2. Minus 2 and minus 2, it will be 0. Again second one, value of box bottom, that is minus 2 minus gap value, that is minus 2. Minus 2 minus 2, it will be 0. Next third one is diagonal value as 0, and if there is a match, that is G and G is matching, so hence it will be plus 1. 0 plus 1, it will be plus 1. Hence 0, 0 and 1, in these three values, 1 is the maximum value. So hence we have to give as 1 as the score in G and G box. Coming to the fourth step, back tracing or trace back. After scoring is done, draw a line from maximum to minimum value with back towards 0. The fifth step or the last step is optimal alignment. Align the pair of sequences after the back tracing. So if you align the sequence for GA, GC or GA, TC with the score as 3. So in the example 2, GAT, CCA and GTG, CCT is for the Needleman-Wunsch algorithm. Coming to the third method of pairwise alignment, that is Smith-Waterman algorithm. So SW is the Smith-Waterman algorithm is mathematically proven to first the best highest scoring local alignment of two sequences. The best local alignment is the best alignment of all possible subsequences or in parts of sequences of S1 and S2. The zeroth row and zeroth column of T are first filled with zeros. The recurrence relation used to fill table T is T I comma j is equal to max of Ti minus 1 comma j minus 1 plus row of S1I comma S2j. Ti minus 1 comma j plus gap penalty, Ti comma j minus 1 plus gap penalty and 0. So a fourth possibility, unlike Needleman and Wunsch, has been added in Smith-Waterman. The traceback starts at the highest score cell in the matrix T and travels up or left while the score is still positive, while in Needleman and Wunsch, traceback starts at the bottom right and ends at top left, which ensures is global alignment. As similar case in the Needleman and Wunsch algorithm, in case of Smith-Waterman algorithm, the match as +2, mismatch as -1 and gap penalty as -2. We first calculate T1, 1 using the recurrence relation. Ti minus 1 comma j minus 1 plus dou S1I comma S2j is equal to 0 minus 1, that is equal to -1. Second step, Ti minus 1 comma j plus gap penalty, that is 0 minus 2, that is equal to -2. Third, Ti comma j minus 1 plus gap penalty, that is equal to 0 minus 2, that is -2. And fourth is 0. So out of -1, -2, -2, 0, 0 is the maximum value. Hence, we set T1 comma 1 to 0. And the next calculate in the similar manner for the other cells. You fill in the whole of T recording the previous cell, if any, used to calculate the value of each Ti comma j. The other cells can be filled and the result will be as shown in the figure. After filling the cells, the final step is backtracing. So in this backtracing, we'll fill from the maximum score to the minimum score with the local regions. So as per the example which we have been provided, like GG, CT, CAA, TCA and AGC, TAA, GG. So the score will be 4. The tools that utilize alignment algorithm. So hence EMBOSS or European Molecular Biology Open Software Suite is one of the software where we can use for the local or global alignments. Applications. Identify conserved interaction pathways and complexes. The second application is functional autology detection. The advantage of the alignment algorithms are the similar sequence region is of the time order and orientation. The disadvantage is slow, memory intensive, cannot be applied on genome size sequences. The difference between global and local sequence alignments are, in global alignment, an attempt is made to align an entire sequence, that is end-to-end alignment. In local alignment, finds local regions with the highest level of similarity between the two sequences. A global alignment contains all letters from both the query and target sequences, whereas the local alignment aligns a substring of the query sequence of the substring of the target sequence. In global alignment, two sequences have approximately the same length and are quite similar. They are suitable for global alignment. In local alignment, two sequences can be locally aligned as local alignment finds stretches of sequences with high level of matches without considering the alignment of the rest of the sequence regions. Global alignment is suitable for the aligning two closely related sequences, whereas local alignment is suitable for aligning more diverse sequences or distinctly related sequences. Global alignments are usually done for comparing homologous genes, like comparing two genes with the same function, that is in humans versus mouse, or comparing two proteins with similar function. In local alignment used for finding out conserved patterns in DNA sequence or conserved domains or motifs in two proteins. A general global alignment technique is the Needleman and Wunsch algorithm, whereas the local alignment is Smith-Waterman algorithm. Examples of global alignment uses embossed needle, needleman wunsch align nucleotide sequence specialized blast. Examples of local alignment tools are blast, emboss, water, and L-align. Coming to clustering, phylogenetic clusterings by simple matching coefficient. A simple matching coefficient or Rand similarity coefficient is a statistic used for comparing the similarity and diversity of sample sets given two subsets A and B, each with n binary attributes. So, simple matching coefficient is defined as number of matching attributes divided by number of attributes. If you say in table, so M00 plus M11 by M00 plus M01 plus M10 plus M11, where M11 is the total number of attributes where A and B both have a value of 1. M01 is the total number of attributes where the attributes of A is 0 and the attribute of B is 1. M10 is the total number of attributes where the attribute of A is 1 and the attribute of B is 0. M00 is the total number of attributes where A and B both have the value of 0. One of the example for multiple alignment, so or clustering. So hence if we have more than two sequences such as seq1, seq2, seq3, ATT as seq1, AT as seq2, ATT as seq3. So first we will make the alignment of the first sequence with second, first sequence with third and second sequence with third. So after getting, we will make a matching coefficient. Seq1 and seq2 has score of 2, seq1 and seq3 has 3 and seq2 and seq3 has 2. So hence after matching coefficient, as 3 is the maximum number, so hence ATT first sequence and third sequence will be having the similarity and the second sequence will be diverse. So hence the phylogenetic tree will be drawn. Coming to conclusion, sequence alignment or sequence comparison is a way of arranging protein or DNA sequences to identify regions of similarity that may be consequences of evolutionary relationships. In general, we can compare two sequences by placing them above each other in row and comparing them character by character. Emboss includes tools for local and global sequence alignments. Multiple sequence alignment or MSA methods refers to a series of algorithmic solutions for the alignment of evolutionary related sequences. While taking into account evolutionary events can be mutations, insertions, deletions, and rearrangements under certain conditions. These are the references used for my presentation. Thank you.